Welcome to this evening's event. It's a really beautiful evening. We're so thrilled about the weather and it's so lovely to be able to do a face-to-face event in the Botanic Garden. This evening's event, as you can see, is a collaboration between GLAM, Oxford Garden Libraries and Museums, specifically the Botanic Garden in this case, and Reuben College. And Reuben College is the university's newest graduate college. You may know already quite a bit about it. We are just about to welcome our first 100 students. They're arriving in the next 2 weeks, which is a really exciting time for us. And as a college, we've got a real vision for interdisciplinary research. And the college is really going to be focusing on how to embrace the challenges of the 21st century, difficult challenges which need people to work together across disciplines. And we're going to welcome students to this, and we've got a brilliant group of students already. I'm Janet Stott. I'm the Deputy Director of the Museum of Natural History here in Oxford, and I'm also an official fellow of Reuben College, so that's why I'm here introducing it, and we're delighted to be doing this. As I've said, the college has got this particular brief, but one really important thing about the college is it's got this partnership, a really unique partnership with the gardens, libraries and museums. No other college in Oxford has got this kind of relationship. And it comes partly out of the building. You can see on our banners here that the college is going to be based in a beautiful building. It's a building which is the Radcliffe Science Library, which is undergoing renovation and making it a library fit for the 21st century for scientists. And also in the basement of the Radcliffe Science Library, there is a huge double basement which used to be used for book storage, and those books from the Bodleian have now gone out to Swindon to a huge new storage. And we're creating an amazing collections and teaching research centre for the museums of Oxford. So there will be world class collections in the base of the college, and then in the basement, and then the college will be on the next 2 floors. So it's an amazing collaboration that we have, and we're looking forward to lots of opportunities of working together between the college and the gardens, libraries and museums. And one of the first things is a seminar series, and this is the second in a series of seminars that we're going to have between members of the gardens, libraries and museums and members of the college. And we're aiming in this to bring to the fore the amazing collections that Oxford has, but looking at their potential for research and for engagement in relaxed conversations. So that's what we're aiming to do tonight. It should be a really nice, relaxed event, and we hope you will take part and ask lots of questions. And we have 2 fantastic people to present to us. So we have Dr Chris Thorogood, who is the Deputy Director and Head of Science at the Botanic Garden and Holker Arboretum, and Professor Emily Flashman from the Department of Chemistry. And I think I will leave you to introduce yourselves. Is that okay? And it's going to be a nature of a conversation between them. It's a very relaxed event. Great.
Okay. All right. Thank you very much. Yeah. So I'll start by introducing myself and why I'm here. So I work in the chemistry department, and I would call myself an enzymologist. So I study enzymes, the catalysts in our cells and in all living organisms that make chemical reactions happen in our cells. So they trigger the reactions of life, so they enable life. And enzymes make chemical reactions happen, and they can do this either fast or slow, depending on the nature of the enzyme. And I'm interested in enzymes that use oxygen. These enzymes can sense oxygen and trigger a response. So if there's a sudden decrease in oxygen availability, the organism can respond and adapt to that decrease in oxygen availability. So we consider that from the point of view of flooding when we're thinking about plants, because when plants are flooded, they experience a reduction in oxygen availability. And so we're interested in how plants adapt at the molecular and the physiological level to flooding. But there are lots of aquatic plants as well, which have all sorts of different ways of adapting to being surrounded by water and don't get damaged by this in the way that some plants do. So it's really interesting to come and have a chat with you, Chris, about how different plants can adapt to living in an aquatic environment.
Absolutely, I wholeheartedly agree. And when we first conceived the idea for this seminar, and we were trying to think where might our areas of research overlap, weren't we? And we struck upon flooding and our interest in plants, both at the molecular and the microscopic, and also the macroscopic level, and their responses, their adaptations, and their responses to abiotic stress, environmental stress, such as flooding. And so we really wanted, we're not just going to show aquatic plants, but we're going to focus on those tonight, hence the paddling pool, in case you were wondering. This is our way of bringing some of the aquatics to you. So we're going to take you on a bit of a journey tonight through some of the research that we do, and in collaboration with other scientists as well, to explore and examine some of those extraordinary adaptations, both structural and functional, that plants have. And also along the way, we'd like to point out some of the potential applications and how they might be relevant to people and technology and our everyday lives.
Okay, excellent. So are we going to start with this mucky thing?
What I might just say actually before we start with the mucky thing, which is more interesting than it looks, and we'll show you in a second. But I also wanted to extend on the welcome that Janet gave to everyone tonight to the Botanic Garden, because that's where I work, and it's a very special year for us because it marks our 400th anniversary. So the Botanic Garden, as you may know, is the oldest botanic garden in the UK, and it was founded in 1621 as a physic garden, primarily to grow medicinal plants to teach students here in the university about the medicinal properties of plants. And it was inspired by some even earlier physic gardens in Italy and elsewhere on the continent, particularly the Botanic Garden in Padua, which is the oldest in the world, founded in the 1500s. And we still have the cruciform layout in the Botanic Garden that was inspired by that. And today we have 5,000 or so different types of plant from all around the world, which is what botanic gardens hold. So botanic gardens collectively, it's been estimated we hold 31% of the world's species of vascular plants. So a vital resource for conservation and research, which we're going to talk about tonight. But I did want to show you, before we show you the other slightly ugly plant, I did want to show you one that sometimes people tell me this is ugly. I don't think it is. It's not something we wanted to focus on in tonight's seminar, but we're just catching the end of its flowering, and I couldn't resist bringing it to show you. It's called a Brim Rape, is its common name. Its Latin name is Orobanchipicridis, and it's actually one of Britain's rarest species of plants. It's protected by law, it's on Schedule 8, and it's a parasite. So this is the plant that we're looking at here, this ivory-coloured plant, and it's siphoning off its food from this green weedy looking thing, which is a hawkweed ox tongue, and it has no leaves or chlorophyll of its own. It steals all of its food. And we're growing it for conservation reasons, because these plants are often neglected and ignored by botanic gardens. But we're also interested working with other scientists. Forgive my poor laminated efforts, but to show you, and we can share these photographs after. This is a relative of the plant I've just shown you, and it's called Sistanki, or a desert hyacinth is its common name. And these are some extraordinary parasites that grow across the deserts from the Middle East through to China. And we're interested, working with colleagues at the University of Reading and in China as well, and the Middle East, to understand more about the relatedness of these plants, which are very confused. But with a view to informing how we might grow such a plant as, for example, a famine crop, because this plant is widely eaten by people where it grows, and it's sometimes used for herbal medicine as well, particularly in China. And if we are able to grow this as a crop, we might relieve pressure from native and rare populations that people might be illegally harvesting, and we know that can happen, and they're traded. And we could actually grow them in conjunction with shelter forests, which are planted across deserts to halt land degradation. We talk a lot about climate change, quite rightly. We don't always talk about land degradation and desertification, which is a major consequence of that. and people are planting shelter forests across the world to try and halt that land degradation. And you can grow this parasite on those shelter forests as an ancillary crop. And so wouldn't it be wonderful if people could grow a crop that we're not growing today that could be a useful famine food? It requires no irrigation or feeding, and it would relieve wild populations from illegal harvesting as well. So that's an example loosely linked to this rather unusual parasitic plant here. And speaking of unprepossessing plants, I think Emily's going to show us, and maybe as she talks, shall I sort of show people so they can get a better look as you're explaining, Emily?
So we're focusing today on how plants are adapted to living in an aquatic environment, i.e. how they cope and can get enough oxygen to be able to survive even though they're surrounded by water. But actually plants started out as being aquatic organisms, and so the very first plants were actually green algae floating in water, and then these developed into filamentous algae, and it was actually not until these Marchantia plants first came out of the water and occupied the land that plants were exposed to the air. So actually these Marchantia plants were, so they're a type of liverwort, and these liverwort plants had to adapt to an entirely different environment. They've been used to being supported by the water and not having to have a rigid cell wall, and not being exposed to too much UV light, and so they had to rapidly adapt to being exposed to this environment. So actually they kind of did the opposite of what we're talking about now. They adapted to coming out of the water, whereas many of the plants we're going to talk about tonight are adapting to living again in the presence of water.
It's wonderful. It's like we were chatting before the seminar started and talking about this as a sort of land conqueror, because 470 million years ago, the relatives of these plants, the bryophytes, the liverworts and the mosses, were the ones that pioneered land and really changed the course of evolution of plants forever by conquering the land. So it's fascinating to hear the research that you and your team do, Emily, at the molecular level on these plants, which feature very prominently in our teaching here to undergrads about the evolution of plants on land.
Yeah, so these types of plants were the first to evolve the types of oxygen sensing systems that we work on, and I'll come back to the molecular details of that a bit later on, but they're a really good sort of pioneering example of how plants developed, and we can learn from that. Yeah.
Actually, Emily, I have a question for you actually that I've just thought of whilst we're talking about this before we move on. I know you're going to talk about a couple of other plants as well, but whilst we're talking about the Marchantia, do you think there's scope in the research that you and your colleagues do to look beyond some of the better known examples in the plant kingdom across the phylogenetic tree, so that the family tree of plants? Do you think there's scope to look at some of the more obscure and what that might tell us?
Definitely. So as long as we've got the genomic information that can tell us what the sequences of these, the DNA sequences are for these oxygen sensing enzymes, then we can predict the structure of these enzymes. We can biochemically look at them and see how they react with oxygen, and start to look at the differences between enzymes from different organisms. And actually we've got a grant application in at the moment to do that kind of thing, so hopefully we'll get funded to do some more of that work.
Fantastic. Yeah. I'd like for us to talk a little bit about a discipline. Actually, it's no coincidence the plant did this, grabbed onto another one. I'd like to talk a little bit about a discipline called biomimetics. In the introduction, we heard how interdisciplinary research is something that is of interest, and it's something I think that is done very well here at Oxford. Actually, we have scientists and other researchers from many different disciplines working together to tackle questions, which I think is a wonderful approach. I'm quite new to interdisciplinary research, so my research originally was using molecular tools to answer evolutionary questions, and since then I've started working with chemists and physicists and engineers. And it's amazing what can happen if you bring together people from disciplines to try and answer questions, some of which are important to all of us. But before we go on to that, and before we explain why there's a paddling pool in front of us this evening, I wanted to show, at the risk of showing you yet another slightly unattractive plant, this, can someone tell me what this is? Because I know someone in the audience must recognise what this is. It's not quite that, but it's similar. Yes, it's a burdock, exactly. So you will find this probably walking around even this evening. It grows all over the place, and I just plucked one. I say plucked, actually. I had to yank it out of the hedge to the amusement of some onlookers. Anyway, this is a burdock. It's very common. The reason I'm showing this is that this was a source of inspiration to an engineer called Georges de Mestral, a Swiss engineer, who it is widely said, it was the inspiration for his concept of Velcro that we all use every day. So we're all very familiar that if we take these fruits, and actually, by the way, I've lost most of them between them sticking to my car and various other things along the way. But you can see the principle of Velcro in action. So these fruits are covered in hooked spines, and these hooks connect to a loop. In this case, the loop's on my cotton, but in nature, it will connect to the fur of animals. And so from a very basic principle, what we've got here is a plant that hijacks animal couriers to spread its seeds as a principle, has been applied in technology to something that we use in our everyday lives. And really that's what biomimetics is all about. It's about, and you'll hear biomimicry as well, it's about mimicking a structure or a function, and in this case we're going to be talking about plants. There are others such as gecko skin and all sorts actually, that we can use in technology. And this is something, I'll take these off now, this is something that that really came of interest to me working in Oxford only relatively recently in my research career, and I find it tremendously exciting. And so I think what we can do now is maybe move on to an example in the paddling pool, which I've been desperate to do. So maybe we'll do that. Before I do, I think in a moment I might invite everyone, if they're happy to, but fine if you're not, to maybe gather round at a distance you feel comfortable with to look at the plants so you can really appreciate their form and function. But I'm really excited that we can show you tonight what I think is one of the most astounding and astonishing feats of botanical engineering. I can't think of something more impressive. We saved for this evening the largest leaf of a giant Amazonian water lily in our heated pond in the glass houses, specifically for this occasion. It's the largest one that grew this year, and that's why it's looking a bit tatty, so sorry for that, but we did want to keep it for tonight. They're starting to decay now. And I hope you'll agree that it's astonishing but also beautiful. It's a structure that is quite mesmerising, and actually when we lift them out of the pool, which we do regularly because they grow so quickly, we have to remove leaves. People actually gasp in astonishment at the beauty of these water lilies. And some scientists that I was working with on a different project, some physicists, we were walking around the water lily pond, and they're like kids in a sweet shop because they don't know a lot about plants. And so they'll say to me, "Oh, what's this?" And I'll tell them. And we came to this, and they said, "How does it work?" And I told them what little I knew. And they asked me some, and it's often, by the way, the simplest questions that I don't know about you, Emily, but I find are the hardest to answer. Yeah. So just to warn you. but they were asking things, why is it so big? And I sort of mustered together an answer that I didn't really believe in myself. And I thought, well, I'm going to go away and explore what we do know. And to my astonishment, we knew very, very little about this until we started working on a project together that now we have scientists working on in New Carolina, Paris, and the UK, trying to solve the 200-year standing mystery of how do these leaves to grow, and why do they grow to be so large. Not only that, how and why are they so strong as well. So I think what I'll do is I'll invite people to come to the paddling pool which is a sentence I haven't said haven't said yet in my career, and to show you the leaf, and to take a closer look for those who would like to, and we'll examine it together. So the things I'd like to point out, and this is a baby, by the way. This is a really small one. So they grow to reputedly up to 3 meters across, but certainly 2 meters and a bit easy. And you'll notice some interesting features. It has an upturned lip, which helps it sort of tessellate and stay afloat. It has strength conferring abilities as well. It has what we call a sinus, so it has this sort of dip here that you'll notice around the rim. This means that if the leaf, strong as it is, becomes flooded with water, there's a point of egress. The water can exit and leave the leaf so that it doesn't flood. It has tiny pores all over the surface, different to the stomata that we think of in leaves. They're tiny pores that actually transmit water, like rain, for example, and dew, down through the surface of the leaf into the water column as well. So what we're looking at here is a sort of multi-purpose leaf. It has these structural adaptations, and Emily and I were saying at the beginning of the seminar about structural and functional adaptations, and this is what we call a multi-purpose leaf. If you flip it over, it's beautiful, as I say. It's covered in spines. They are absolutely vicious. They will tear through your flesh, so please be mindful of that if you touch it. We wore gloves to get it here. And this protects it, of course, as you'd imagine, from herbivorous predators under the water. I brought an ordinary water lily to show you the size of a typical water lily. This is quite different, I think you'll agree. But it also has quite a unique venation pattern. So it has these sort of rather like spokes of a wheel, and then at right angles to those, additional spokes. And so working with these physicists, and I won't go into the detail, and can't actually go into the detail of some of their very complicated mathematical modelling, but we did a series of experiments to see the load-bearing capacity and see how much weight it can sustain, how it deforms, so what shape does it take as it deforms, and then all sorts of mathematical modelling to understand its load-bearing capacity. But what I will say is you'll often hear or read or maybe even see on the internet that these giant leaves can support the weight of a small child, and I want to be clear that is not a myth. It is true. And it's happened here at the Botanic Garden. People have brought their toddlers in and put them on the leaves. And now this is I'm going to caveat this experiment with the fact that this is a small leaf, so it won't carry as much as so these are, I believe, 1 kilogram weight of sugar. Emily, helpfully, I asked her before the seminar, I said, what's the weight of a newborn baby? Because I don't know. And you knew exactly, because we Googled it. And so what?
3.5 kilos.
3.5 kilos. And there's 6. So we've almost got two babies' weights worth on the leaf. And I can tell you, we could comfortably put another couple of kilos on there, but I didn't want to rob people of the local Sainsbury's of all of their sugar, so I didn't. But I think you'll agree that it has quite some strength in that leaf to be able to sustain that weight. So if we think to where this plant grows in nature, and that's what Emily and I are doing in this seminar, is sort of talking about some of these principles. But then if we think, if we contextualise that with how and where these plants evolved, these water lilies grow in the Amazon. I haven't seen them in the wild. Maybe people here have. But interestingly, what I found in doing this research is that they don't and they don't mainly grow in the Amazon proper, as we might think of it. They tend to grow in lakes, sort of oxbow lakes and fast-drying pools around the Amazon. And so that got us to thinking, perhaps these leaves are responding to an environmental stress not only of flooding. They have to grow very, very quickly and get their leaves out as fast as they can, outcompeting all the other things that might be growing there, so that they can maximise their surface area for photosynthesis in an area that's not going to last that long. They don't live for very long, because these pools will dry out and then the plant will die, and then the whole thing happens again. And so they're growing very, very quickly. So I think when we're talking about the strength and the structure, and we're thinking about the habitat, we can imagine that this is a plant that has evolved this suite of structural features to outcompete other plants very quickly in a certain environment in which it's grown. I don't know if Emily, if you want to say anything else at the moment. If not, I'm going to move on to a couple of other examples.
No, that's fine. I was just going to ask a question. That's all right. So if there's a lot of rain, so this is its photosynthesizing area, this big leaf, and so that's how it's going to generate as oxygen as energy, and presumably then if this gets flooded, then the carbon dioxide can't get in for the photosynthesis, and so having that egress is really important for the water to come out.
Exactly. No, quite so. And the truth is, we don't have really much empirical data to know any of these things, but I would certainly infer that. Yeah. I'm aware that we might be blocking the camera, but I do, whilst everyone's here, if it's all right, want to show a couple more examples whilst we're all around the paddling pool. And the next one I'm going to show, so we talked a little bit about applications potentially in technology, and I want to show another botanical gem from our pool in the glasshouse. Now look what happens when I try to push this under the water. You can see it's having none of it. So this peculiar plant, which is called a water lettuce, or a pistia is its proper name. In fact, what I might do is pass one round and have people, if they want to, they don't have to feel the surface, and maybe someone could describe what they think it feels like or reminds them of. Or rubber gloves, okay. Guinea pig ears. Yeah, I can see that. Nice descriptor there. Velvety. That was a word I was thinking. Yeah. So you'll notice when you feel it that it does feel very, very velvety, and that's because it's covered in a surface of very tiny hairs and very dense hairs that grow closely together, and they form an airlock around them. And what this means is that the structure, actually I can show you what that would look like under a microscope. It would look something like that. Well, it would look exactly like that, actually. So these tiny hairs that grow in close proximity, and they trap air. And what this means is that this surface is selectively absorbent, and so it means that it can absorb a non-polar liquid, such as the olive oil from the kitchen cabinet in the Botanic Garden, but it won't absorb water. So at the risk of making a real mess of this, maybe I'll do it on the grass. If I pour the oil on, it will actually absorb very well, and it won't even come off. So I know that's probably difficult to see for most people, but the oil has gone straight through into the surface. But as you saw, not so the water. And why might this be of interest? Well, because if we were to have an oil spill, which is an environmental disaster, there are some scientists in 2016 who suggested that if we were to make a synthetic replica and we were able to industrialise and commercialise such a synthetic surface inspired by this, we might be able to soak up oil from an oil spill. And that's wonderful, because what we don't want to do is pump chemicals to disperse oil or burn it and cause secondary pollution, in some cases perhaps worse than the original oil spill itself. So I think that's a wonderful application. And what I think is particularly beautiful about this idea is that this particular plant is a pan-tropical invasive pest. So if you go to tropical waterways all around the world, it chokes waterways. It's been introduced by people to places that it has no business in being, and it causes problems. And it wouldn't be wonderful if this plant that we have introduced where it shouldn't and is a problem could also be the solution to another problem that we've created. I think there's something quite nice about that. And then I think I'm just going to show one more around the paddling pool before we sit back down, and that's a similar one. Does anyone know what this leaf is? This is a sacred lotus, and the reason it's called a sacred lotus, by the way, is because it grows in muddy tropical water systems, and its leaves, unlike the leaves of other plants, look very unblemished and free of dirt, and therefore pure, and so it's said to be sacred. And there's a very good reason for that. So again, if I pour water on here, I think you can see just how repellent this is. It doesn't last for long. And so, yes, this leaf is waxy, which you might have guessed, but then So are all leaves, because they have waxes to protect them from desiccation and water loss. But there's something else about this leaf. So this is an image that someone kindly said I could use and show. And what you'll notice underneath those water droplets are those little warts or papillae, because this surface has a texture that you can't feel it and you can't see it, but it's covered in microscopic tiny little beads. And this double structure of soluble lipids or waxes in a matrix in which we have these beads set, what this does is it confers a certain level of cohesive force that causes water droplets to form spheres or beads quite distinctly. And that reduces their amount of surface area to cling, and so they roll off. And so the slightest breeze, or even just the topography of it, the shape and angle, means that the water just beads straight off, and so it doesn't linger there at all. And as it goes, it carries dirt with it, and so the clever illustrator who made this has actually added some dirt, and you can see. And so beads will roll off the surface of the leaf, and they will carry dirt with them. So this is a self-cleaning leaf, and this was the source of inspiration for many self-cleaning surfaces that are used today in glass and paint and other such surfaces as well. I think maybe we'll let people sit down and we'll move on to the next plant.
So presumably then, for both of these plants, those structures on the leaves allow them to survive in their aquatic environment because they allow gases to get into the leaf while preventing water from occupying that space.
That's exactly right, Emily, because the one that we just looked at, particularly the sacred lotus, it's no coincidence that it looks so clean because, and I don't know if there is data to show this, but my assumption, like yours, would be that if it's a clean leaf rather than one covered in a muddy film, then surely it's got better access to light and therefore can photosynthesize effectively. And so it's got a hardwired optimization for capturing light, just as in a different way the water lily did.
Yeah, cool. Okay, what should we talk about next?
Do we want
rice. Should we talk about rice?
Whilst we're talking about aquatic plants.
Get a bit molecular. Okay, so we've talked so far, Chris has talked so far about plants that are adapted to surviving in an aquatic environment. But most of the plants that you'll be familiar with, and including the crops that we eat, are not adapted to that. They like to survive in a dryish environment. They require water, but if they get waterlogged or flooded, then they can't survive for very long. And that's why flooding is a big and increasing problem when we think about food security and crop production, especially with the onset of climate change. So thinking about how plants cope with being flooded and thinking about whether there's any way to help them tolerate flooded conditions a bit better or for a bit longer is something that a lot of people are focusing on. And we've learned a lot from rice. So this is the rice plant. And rice, as you're probably aware, can grow in an environment where it can tolerate a certain degree of flooding or waterlogging. And because rice are known to be able to tolerate this, they're a good example to understand the molecular mechanism that underpins how rice can tolerate it. So what rice will do if rice becomes waterlogged is they'll have one of two adaptive responses. They'll either basically turn off their metabolism to conserve energy, so that when they become flooded, then they're not getting oxygen, they can't produce energy. So by shutting down their unnecessary metabolic processes, they can kind of sit and wait. So they just try and turn everything off and sit and wait and see if they can do that and survive slowly through anaerobic metabolism on their starch reserves. And hopefully they won't run out before the water levels recede and they can get oxygen again and start to grow again. But the other mechanism that different varieties of rice use is an escape mechanism. And so what they do in that instance is, instead of turning themselves off, they rapidly grow above the level of the floodwater and form some structures called parenchyma, which are essentially snorkels. So right down the stem of the plant, these snorkel features will form, and these snorkels will allow oxygen to get down the plant and access all the tissues that need it. And so they can keep on growing, and so in that way they can survive the flooding event. And what scientists did probably from the mid-2000s is use these two examples and worked out how these responses were underpinned at the molecular level. And I'm going to get out my diagram now. So what we have under normal oxygen conditions, here's our rice plant growing quite happily. And they found that there's certain transcription factors, okay? So they're called ERFs, and they are transcription factors. And what these do is they turn on genes. Now, under normal oxygen conditions, these transcription factors called ERFs are degraded. So they're produced, but they're degraded by the plant cell. But under low oxygen conditions, they're stabilized. And so once they're stabilized, these transcription factors can induce the expression of genes which will allow the rice plant to adapt. And so those genes will either be to promote growth, this rapid growth, or to change the metabolism to switch from the rapid aerobic metabolism to the anaerobic metabolism. And the really clever thing, this is the bit that I like, the oxygen sensing enzymes, is that we have these enzymes and they use oxygen. So under normal oxygen conditions, when there's no flooding, these enzymes use oxygen, tag the transcription factors with a little molecular signal, and that sends it for degradation. But when there's low oxygen, these enzymes can't work efficiently because there's not enough oxygen available. If you remember at the beginning, I said enzymes can work fast or slow. So here they're working fast, and here they're working slow, and the transcription factor is stabilized to induce the expression of these flood tolerance genes, and the rice is able to survive due to these mechanisms. Now, there's a really great story from rice because one particular earth transcription factor in some fairly low producing but very flood tolerant a very flood tolerant form of rice called sub 1A is actually inherently stable, so it doesn't undergo this process. And so what scientists did in the late 2000s was transfer the gene for this sub 1A, this particular earth called sub 1A, into more commonly grown rice varieties. And that extreme flood tolerance that they'd seen in this rare rice variety was then conferred into all these more commonly grown rice varieties. And actually that's been a tremendous success in improving the flood tolerance of all these engineered rice varieties that they've developed. And so I read a figure that in a flooding year, then farmers are seeing a 46% increase in yield compared to the rice varieties they were using before, but without any kind of detriment in yield in a normal year. So that's a really amazing success story, but it's quite unique to rice because they managed to find this one particular earth transcription factor that wasn't destabilized. So what we're doing, the research in our lab is we're trying to understand these enzymes and find ways to change the rate of activity of these enzymes with oxygen so that we can shift this pathway over in this direction, stabilize these transcription factors, and confer the same kind of flood tolerance in not just rice, but in all sorts of other crop plants. So that's the kind of thing that we're trying to do. So it's really interesting to look actually at these enzymes from different plant species which come from different environments and see how they react differently with oxygen. And so we've looked at the enzyme from Marcantia and seen that its sensitivity to oxygen is actually really quite different to the sensitivity of, for example, rice enzymes, or what else have we looked at? Maize and Arabidopsis, which is the sort of common weed that people use as a plant model. So that's the type of thing that we're really interested in doing, and learning from these different plants that have different tolerances to oxygen is really exciting.
Emily, one of the things I was thinking as you were speaking was we were talking about some of the potential applications of some of these plants that we're looking at. And I wonder, I'm not sure if this is a gross oversimplification, but is it fair to say that an application of this very detailed molecular work that you're doing is linked to trying to grow crops for a growing population in a changeable climate? And so if we frame it in that way, do you see your research in that way, Emily?
Yeah, because it will allow crops to be grown in environments that might otherwise have to be neglected because they're too susceptible to floods and, well, you know, and drought and all sorts of changeable climates. But it's one aspect of that, yeah. So that's what I hope we might be able to do one day.
My other question, whilst we're talking about the rice, was your thoughts on looking at wild crop relatives. We grow some of those at the Botanic Garden, and I guess coming back to a similar question I asked earlier about, you know, looking at Marcantia and rice, but what your thought, because you did mention other species there, and what your thoughts are about future directions.
So I think that's really important, because that's, for example, how this whole the rice that flood tolerant, that's how they were discovered, by looking at this fairly rare rice variety which has this particular flood tolerance. So it's really important, and we can learn a lot from it. But you can hear the but in my voice, right? I think it takes a long time. And I think that we need a dual approach of both learning from nature, so looking at different plants, different varieties from different environments, but I also think we need to do a bit of rapid human-driven evolution, by which I mean basically engineering enzymes in the lab to try and take those two approaches to find enzymes that can be optimal for oxygen sensing and flood tolerance.
Understood.
Yeah.
Understood. I can see that we're pushed for time, so I'm going to come on to my last my last specimen that I wanted to show, which in many ways I think is equally beautiful to the water lily, if not quite as impressive in terms of size. I'm sure some of you will recognise this. It's a carnivorous pitcher plant. Its proper name is Nepenthes, and they originate predominantly in Southeast Asia. And I've just plucked this one. There was already an insect in it, I've noticed, which I didn't put in. I'll explain briefly how it works and then why it's important. This is a leaf in evolutionary terms. It's not a flower, and it's not even a leaf appendage. This is the actual leaf. And it's a vessel, so it's an epiacidiate leaf, so it's fused to form a vessel. If I tip it upside down, liquid will come out. I didn't put that in. Those are its digestive enzymes this one opened a couple of days ago, and it opened with those enzymes in there. You can think of this as a veritable plant stomach, if you will. It produces nectar, much as flowers do, that's sweet and sugary, and attracts insects. They investigate, looking for a reward. And what happens is they slide off this stripy, slippery structure at the top here, and they tumble into the leafy abyss, where they drown in a pool of digestive juices, and they release nitrogen. I can see people wincing. They release nitrogen and to a lesser extent phosphorus that benefits the plants. And these plants evolved in environments where nitrogen is very scarce. They grow in acidic, waterlogged environments or rain-leached environments where nitrogen is washed away and there's not much for the plant. So even though this is, in energetic terms, for a plant to produce an expensive investment, it's well worth doing because it means that it can get the nitrogen it needs and outcompete its neighbours, which of course is what these plants are all doing over a course of evolutionary time. So I was long interested, well, I've always been interested in these actually, but even from a research point of view in the evolution of these plants, which is a whole different seminar. Again, working with other scientists helped open my eyes to understanding it from a different perspective. So again, those quite straightforward questions, well, how does this work? How does this work? Someone once told me the 5 whys if you ask 5 times and you get to the point where you don't know, and I didn't. And so we explored together, I and some physicists explored this slippery rim, which is fundamental to the trap, because this is the part that the insects aquaplane off, which another colleague of mine works on in Bristol. And my clever colleagues made soft polymer replica models of this peristome. They did mathematical modelling, they did, you name it, they did it, to understand more about this structure. Meanwhile, a student of mine looked at the structure under scanning electron microscopy, and what you may be able to just about see is that it's made up of ridges, and those in turn are made up of smaller ridges of what look like roof tiles, basically. That's basically how the structure of this works. And we did a series of experiments where we positioned both insects, which is the natural prey, but also oil, which is a foreign droplet if you're a physicist. And we watched the behaviour and how it moves in some detail. And what we found is that things move on this peristome in a way that is much more controlled and driven and less arbitrary than you would expect on a normal surface. So if you're an oil droplet, you're guided exactly in the right direction. If you're an insect, if you land on a certain point, again, you're guided along these chutes or energy railings down into the trap. So this trap knows exactly where it wants those insects to end up. And so when I was a child and I used to read about these plants as a boy and I was fascinated by them, the narrative was that these are sort of sitting there, passive traps that insects happen to just tumble into and then the plant benefits. That's not the case. It's much more elaborate and beautiful, I'd say, than that. And these peristomes are driving these insects in a very controlled way. And what I'm now doing with some mathematicians here in Oxford at Mathematical Institute is to look at a variety of different shapes of these peristomes. And this is the wonderful thing, I love this about Oxford. Our student who's just finished has been generating risk heat maps of peristomes to see where it's a whole new meaning for risk assessment, where it's worse for you, where you're worst off landing as an insect because you're most likely to fall in, which is very exciting. What's the potential application of some of this? Yes, it's exciting, it's interesting, and I think everyone can appreciate that, or I hope. But it's also relevant to us. So there is a branch of technology called slippery, liquid, porous infused surfaces, which is a form of technology, rather like the self-cleaning surfaces that we've looked at already. But this suffers, I'm not an expert on this, but I understand that it suffers from a lack of control of movement of liquids, which of course we've just potentially solved by talking about not just our work, but the work of other scientists by talking about this peristome. But also the movement and manipulation of small volumes of water is quite useful in technology in many senses. So our inkjet printers at home, refrigeration devices, medical devices, they rely on microfluidics and the movement in a controlled way of small volumes of water. So wouldn't it be wonderful if we can inspire technology based on a carnivorous pitcher plant? And wouldn't it be wonderful as well if we had floating solar panels on the ocean that were an economy of material like the water lilies are that we saw? Because an economy of material is what they are. The lamina, the leafy bit of this water lily is really not much thinner or thicker, it's the same thickness as that of the giant water lily, and yet that one gets to grow very, very big. It's a very efficient use of biomass to cover a large area, because that's what the plant evolved to do, and so that could be a form of inspiration. And the risk of going on horribly beyond the time, I wonder if I might just finish with a picture then, which speak 1,000 words, of a peculiar little plant that we described. And I want to make clear that I don't take the credit for this discovery at all, even though we described it as a new species. There is a rainforest explorer, is what he calls himself, called Dome Nikong, who lives in the rainforest in Malaysia, and he found this plant, brought it to the attention of a Malaysian botanist, who brought it to my attention, and we've just described it as a new species. And at the risk of sounding pessimistic, but I think it's important to be clear, it's critically endangered. It might already be extinct. So at a time when we're still discovering plants like this one this year, we're losing them, and we're losing plants faster than we can discover them, which is hard to make sense of and deeply disturbing. And all these wonderful applications in technology and for good, let alone the other things, will be lost with them. And so I think a message that I wanted to share as part of this seminar was hopefully that through our work we've shown plants in a slightly different way, but in a way that maybe it will change people's perceptions of plants, because some people, I'm sure not the people here tonight for our seminar, but some people think of plants as boring or inanimate or not relevant to people. And they're highly relevant, and I think we can, as scientists, we have a responsibility to help engender and foster people's care and appreciation for the plant world at a time when it's very much needed. Would you agree, Emily?
I absolutely agree, yeah. We rely on plants, absolutely 100%, not just for food, but for the environment that we live in. So that's a very important message. Yeah.
Thank you. Thanks.